Hi everyone, this is Mr. Sinti, and I want to talk to you about the whole workflow of our class Conservation Biology and some of the questions that we have in the course. Well, as you might know, our organism that we're studying is this wonderful coastal redwood tree, and it's indigenous or unique to California, and it lives right around on the coast. And we're curious about the tree, and for many reasons. One of the reasons that we're interested in the redwood tree is that despite the fact that most of these redwood trees are saved in national parks and in state parks, there's still possible um, vulnerabilities to these trees because of the fact that they're so few in numbers and their biodiversity may not be as great as we would hope. And so when you look at a tree, when I look at a redwood tree, it's very beautiful, but when I look at trees, they look very similar to one another. And so in class, we're going to attempt to look at the differences between trees genetically. And so just like people, we can do um, a look at their DNA, and we can look at a particular region of DNA called a gene, and we can actually see that there are differences between one tree and another. And so a couple things that we're going to consider is that the redwood tree itself, we're going to be able to extract the DNA and look at a gene and sequence it. And we're going to do that by cloning and sequencing, and that's what this video is all about. What's interesting, if I could start off with this, one of the interesting things about a redwood tree is that um, when a redwood tree grows, it can grow either asexually or sexually. And sexually is the good old-fashioned way with cones, and the seed comes down from the tree, and a new tree will grow from that seed, pollen and egg, and it's sexual reproduction. But the tree can also grow from these growths down below called redwood burls, and so new trees can grow. And what's interesting, this is considered to be asexual, meaning that it's just simply the daughter tree is simply an offshoot of the parent tree. And so we believe, and I say we believe, that this tree right here is identical to this. And so if it is identical, they should have the exact same DNA. And so one of the things that we're looking at in class is actually sequencing a burl or a daughter of the parent tree, and we're going to consider that. Another thing that we're interested in is some redwood trees, as you can see over in this picture, are albino, meaning that they don't produce any chlorophyll. And this is odd because you need chlorophyll to live. And if you don't have chlorophyll, you're not going to be able to photosynthesize, and you're not going to survive. And it's like, well, how can you have a albino tree then? Well, if I can continue this a little bit, if this albino was connected to this tree, it would then use the sugars produced by this original tree in photosynthesis, and the sugar would pass over. And so basically this tree is supplementing the deficiency of the albino tree. So here's our question. If this is part of the original tree, then how come the albino is even white in the first place? Our question is, does the albino share the same DNA as the parent tree? It's like, how are we going to do that? How are we going to compare DNA in trees, this tree and the burl and the albino and other trees? How's all that going to work? And so that's what this slideshow is really all about. First thing I want to say is that in order to get the DNA, we're going to have to slice off a couple of needles. But we don't need very many because each needle is composed of many thousands of cells. And a typical plant cell looks like this. Here's the cell wall, the cell membrane. As you may remember from biology, most of the DNA is located here in the nucleus, or genomic DNA. It's located here. There is some DNA in the chloroplast, and there's some in the mitochondria. What's interesting, I want to point out something, that though the DNA is located in the nucleus, let me sort of emphasize it like that, what we're going to be considering is the mitochondria, because as you may remember, the DNA produces RNA, and the RNA travels out to ribosomes, which are these little dots, and they make proteins. One of the proteins that we're going to consider is a protein involved in the process of cellular respiration, which occurs in the mitochondria. What we're interested in is the gene that makes the enzyme for cell respiration, and that's what we're going to use to compare the trees. So a close-up of that notion. Remember DNA up here, DNA makes RNA and RNA makes the protein in the cytoplasm. So say this is a redwood cell, this is a redwood nucleus. What we're going to be interested in is an area, just one small area. The redwood's got thousands of genes, but we're going to look at just one gene in the whole sequence. And that's going to make messenger RNA, and that's going to make go to the ribosome, and it's going to make a protein. And you're like, well, what protein? What gene is it that we're going to study, and what protein is the result of the gene that we're going to study? This is our question. Well, the gene is a gene for this protein. It's a protein that acts as an enzyme in cell respiration. And cell respiration, as I mentioned before, occurs in the mitochondria. And this enzyme is kind of a mouthful, but I'll say it. It's glyceraldehyde 3-phosphate dehydrogenase. And you're like, oh. But it's actually, the acronym for it is GAPDH. And so the protein is GAPDH. We're going to be interested in is the gene on DNA that codes for GAPDH. And so if a tree has the exact same gene for GAPDH that another tree has, we can think that they're identical or clones. And if they're different, then we'll know that they're two different trees. And it's like, what's the point? If we can determine the genetic diversity of the forest, we can get a better understanding of how related the trees are to one another. And the thought being that if the trees are more unrelated, in other words, if the sequences are more different, then the forest might be healthier because the diversity lends itself to being able to be more responsive to diseases that might be coming through the grove or climate change. Because overall, we're worried about the trees. They're not completely saved, and so we want to make sure that there's diversity there in the woods. How are we going to do this? How are we going to look at this gene? Well, we're going to actually extract the DNA. We're going to, it sounds a little tricky, but we're going to make many copies of it, and we're going to put it into a plasmid vector, and we're going to clone it, and then we're going to sequence it using bioinformatics. So let's talk about that. So here's a close-up of the cytoplasm and the mitochondria of a redwood cell. I won't go into the details of this, though I could. One of the things about cell respiration is it's broken up in 3 phases, and so we're looking at just this area called glycolysis where glucose is split in the cytoplasm. If you look close, there's 3 hours here, but there's really 10 steps. Glucose that's made in photosynthesis is broken down in this pathway, and it's split into these 2,3 carbon sugars, and one of them is glyceraldehyde 3-phosphate. The next slide. This enzyme right here, triose phosphate dehydrogenase, or glyceraldehyde 3-phosphate dehydrogenase, is the enzyme that we're considering. And we're interested, remember, in the gene that makes that enzyme. All right, so how are we going to do this? How are we going to look at just one gene out of the thousands that the redwoods have? And the answer to that is we're going to use a biotechnology tool called polymerase chain reaction, or PCR for short. And it's one of the best ways in which we can get a large quantity of DNA to be replicated very fast and efficient. And we don't really even need a cell to do it. And so we're going to replicate DNA without cells. And it's like, oh my God, how are we going to do it? We're going to amplify it. And so let's talk about this just for a second over here. Let me actually come over here and close the slideshow and try to come over here and draw something. And so what we're going to do is, here's all of the redwood DNA. And we're kind of interested in just, let's say, this area right here. And so this little area right here is what we would refer to as a gene. And so we're like, okay, we want this. And we want many copies of this. And so what we're going to do is, curiously enough, we're going to take the DNA and put it inside of a test tube. And so there it is. And this is the template. And so how can we get DNA to replicate it without a cell? Because from our previous discussions, it involves many enzymes and like, oh man, well, how are we going to do it if we're just going to put DNA in there? Well, we're going to need to add a few things clearly. So let's make a little list of things that we're going to need to add. Number one being the DNA from the redwood tree. So that's the DNA or the template. And then what we're going to need to do is add an enzyme called DNA polymerase. And we have this. We're going to add that because that is what's going to replicate the DNA. And it's like, well, how can we replicate DNA without any dNTPs? And so we have to add some dNTPs. And so we can add DNA. We can add DNA polymerase, dNTPs. We're going to add, and I'll mention the details of this in a moment. We can add some magnesium and some buffer to maintain pH. And then most importantly, we're going to need to add some primers. Now let's talk about that just for a second. Primers, 2 of them. Primers are these little pieces in a cell of RNA that attach to DNA to help DNA replication. But since this is not in a cell, we don't really need to use RNA, because if we did use RNA, we would have to add the enzyme RNA primase, and we don't want to do that. So we could just add DNA primers. Yes, can we do that? Yes, we can. So if we added one there and one there, and these would attach here and here and here. And so what's interesting is DNA polymerase, you would think it would want to, let me emphasize it with color. Let's go yellow. You would think DNA polymerase is capable of replicating all of this DNA. It is. But it can only attach to an already existing piece. And if we can add primers to the forward and the reverse direction of this one target area, then replication will only occur in this area. And so, as a consequence, if you let this go over and over and over again, you'll get many, many, many copies of just this one area that we're interested in. And so this is called polymerase chain reaction because it involves DNA polymerase, and it's a chain reaction because it's occurring over and over and over again. I want to talk about that. There's going to be another slideshow which emphasizes it more fully, but it certainly needed to have been mentioned at this point. So let's go back to the original if I can. Go back to the original slideshow on here. Here's our template DNA, and it's like, okay, notice how it's opening up. And if you remember before how DNA opens up, it usually involves an enzyme called DNA helicase. But we don't want to use so many enzymes, and it requires kind of a lot of money to do that. And so one way to open the molecule is simply heat it. It takes quite a lot of heat, so it's about 95 degrees Celsius to do that. But if you apply a tremendous amount of heat, the molecule will open up, as you can see right here. These primers will then attach, or anneal is the word I'm looking for, attach to the target area that we're interested in. Now this is really huge. DNA, it's very long. We're just interested in this one area. Now, it takes a tremendous amount of heat to open the molecule, but then we want to cool it temporarily so that the primers have a chance to attach. And then sort of the third phase of this process is that DNA polymerase will do its thing. It'll grab a hold of the primer and it'll start replicating DNA with those new dNTPs that we have here. And so after one full cycle, open the DNA, primers anneal, extension. How many molecules of DNA? 2. And then same thing, we'll do it again. We'll heat it, and this time the molecule will open up. We'll get extension, we'll get the anneal, the extension, and then we'll have, after 2 cycles, we'll have 4 pieces, and then 8 pieces. And so what you're seeing is that DNA replication is exponential. And so all of this is being done. If I can, I don't know if this is would be very clear, but all of this is being done in a test tube, although you can't see it. But what's interesting about this is, like, you may say to yourself, this could be a lot of work. Okay, it's one thing adding the DNTPs and the polymerase and the primer and all of this, but what's the story with how long does it have to be hot? How long does it have to be cool? And then this is warm. The enzyme works well under warm environments. Do you have to keep rotating this? Do you have to keep like hot, cool, warm, hot, cool, warm? The answer to that is yes, but it would be very laborious, truly, if you had to do it on your own. And so we have a machine that's actually going to do that. But before I get into talking about how PCR can make many copies in just a few hours, and I mentioned it already, heating, cooling, and the question is, how do you even get the DNA from the redwood in the first place? And so that's going to involve DNA extraction. So we're going to go to the woods, take the needles, basically add a bunch of chemicals called extraction buffer, and we're going to crush it in the mortar and pestle, and we're going to pull the DNA out and then purify it. And then what we're going to do is put it into a little test tube on this rack, and then this goes into this machine. This machine looks complicated. It's called a thermal cycler, but it actually just changes temperatures for as long as we want it to. So what it does is it cycles between very hot, cool, and warm. Hot, cool, warm. And you can program it using these buttons down below, and you can get it to do anything you want it to do. And so it's called a thermal cycler because it cycles between these temperatures. And so what we're attempting to do is pull the DNA from the redwood, make many copies of it in a test tube using PCR, and then what we're going to do is actually splice it into bacteria. And so we're going to try to genetically engineer the redwood plant DNA into bacteria. And you're like, wow, what's the story with that? Well, first off, that ability to do that, to combine one organism's DNA with another, is called recombinant DNA. And the reason that we're doing that is bacteria are famous for growing very quickly. And so we could put DNA into the bacteria and let the DNA grow and grow and grow, replicate more plant DNA and clone. And then when we have a lot of DNA, then we can break the bacteria open, pull the redwood DNA out, and sequence it. And so the thing about bacteria, as I mentioned before, is they grow really fast, and it's kind of easy to grow them. Let me go back for a second. So just the point on this is that we're going to have redwood DNA. Whoops. It's RNA. We're going to have redwood DNA. Let's do this. DNA. Redwood. And we're going to put that into a bacteria. So each of them are going to have some redwood DNA in them, and then they're going to grow and grow and grow. And then we're going to sequence the redwood DNA after doing that. So how is it that we're going to do that? Well, let's mention one thing about a bacteria. Bacteria has just one chromosome, and then they have these small little sort of rubber band pieces of DNA called plasmids, and they actually have many of them. Only one is shown. It's a cartoon drawing of that. It's in a circle. Actually, this is a real transmission electron micrograph of a plasmid. We're going to actually, what we're going to do in class is we're going to take the redwood DNA. Let's go over here and do redwood DNA. Let's make a little piece of it like this, and we're going to put it into the plasmid. Now, the plasmid, as you can see, is a circle, so this might pose a problem, but not so much. We're able to cut the plasmid open using an enzyme, cuts it open, and then sort of, let me draw it like this. So we have an open loop like this, and we're going to take the redwood DNA. I mean, you might be surprised that we can do this. I am a little bit also. But we're going to splice the redwood DNA into this plasmid, and then we're going to put this back into the bacteria, and the bacteria are going to grow. And as the bacteria grow, they replicate their DNA and all of their plasmids, and what they do for us is replicate the Redwood DNA along with their own genes. And so that's pretty cool. And so how is it that we cut plasmids, and how do we put DNA into other? We use a special class of enzymes called restriction enzymes that cut DNA at specific locations. Where do we get these enzymes that cut DNA? It's a long, interesting story, but I won't go so far into it. But bacteria possess these enzymes to cut DNA. I believe it was discovered in the 1970s, fairly recently, that when a bacteria is being invaded by viruses, that's what this picture is, the one simple way in which they defend themselves from a phage is that they have these enzymes that cut DNA. And if they can mark their own DNA, the enzymes will simply cut virus DNA, and it's a way in which they can survive. And so scientists, I think at MIT, discovered these restriction enzymes. They're called that cut DNA because they cut only at specific sites. And we've been able to extract them and use them. And it's been one of the most important discoveries of all in biology, because enzymes that can cut DNA allow for this recombination process to occur. And so they cut at a specific area called a restriction site, so they can make a cut. And so you can place one piece of DNA into another. As you can see here, the enzyme would cut it, and then you would use another enzyme, and this isn't a detail because we're actually going to be doing this. We're going to be using this enzyme called DNA ligase, which actually seals the piece in. So we're going to put a Redwood gene into a plasmid and then use the enzyme ligase to seal it. And sometimes it's simply called, we're going to ligate it into a plasmid. And so here's an overall diagram of this process. Here's the plasmid, although it doesn't quite look like it. Let me see if I can help that. The plasmid would be a ring. We cut it with the restriction enzyme. It opens up like this. And then this area in black, let's just say that this is our Redwood gene that we amplified. We're going to put it in, and then how do you get it to attach? DNA ligase. So DNA ligate, so ligate it in. So here's our little rubber band with Redwood DNA in it. Pretty incredible. And then, what are we going to do? We're then going to use the plasmid as what we call a cloning vector. We put that plasmid in to the bacteria, and then the bacteria use it to replicate, and so it's referred to as a cloning vector. The bacteria then carry the recombinant plasmid and reproduces along with it. And then we just simply, maybe not simply, but we grow bacteria on agar plates. We're going to be able to grow this bacteria, and if it grows, as you can see here, these bacteria that are growing on this petri dish with agar actually have Redwood DNA in them. And then the final thing that we're going to do is break the bacteria cells open, purify the plasmid from all the other bacterial DNA that they have, and then we're going to send the purified plasmid into a biotech company, and they're going to assist us in the sequencing. And then that's going to come in email, and we're going to, on the computer, be able to see the sequence of the various redwood trees that we're looking at, and then we're going to make those comparisons. So I hope you enjoyed a brief look at the workflow in this wonderful class called Conservation Biology of the Redwoods. Thanks for watching.